Good morning, good afternoon, and good evening to all of you. Welcome to a new Global IMMUNOTALK. I'm Carla Roslin, and I'm thrilled to reconnect with you after the break and to introduce our Global Immuno speaker today, Dr. Gitta Stockinger. So Gitta is a native of Germany, and she obtained her PhD in biology at the University of Mainz, and then did her postdoctoral studies in London, Cambridge, and Heidelberg. After her postdoc, she became a member of the Basel Institute of Immunology, and she stayed there till 1991. I understand this was a very, very special experience. If you remember, we also hosted Marco Colonna, who also spent some of his formative years there. So I think Marco and Gitta were colleagues at the time. And this was quite special because she could follow everything she wanted, and she had all the support to do that. After that, Gitta became a group leader at the Division of Molecular Immunology of what is now the Francis Crick Institute, and she is currently the Associate Director at the Institute. Now, Gitta's laboratory has been at the forefront of T-cell biology. Her studies have significantly contributed to our understanding of the role of T-cells in tolerance, as well as the role of T-cells in inflammation and infection. And we have particularly learned a lot about the role of Th17 cells, and more recently, about the role of the Aryl hydrocarbon receptor as an environmental sensor of the immune system. Because of her fundamental contributions, Gitta has received numerous awards and recognitions. I would like to just mention that she is a fellow of the Academy of Medical Science, an EMBO fellow, and also a fellow of the Royal Society. I've also learned that Gitta is part of a cycling group of women immunologists, and hopefully she will be going in her fourth trip soon. I really hope that happens and you can enjoy that trip. So I think we are in for a treat. Thanks, Gitta, for accepting our invitation to present, and we very much look forward to your talk entitled Regulation of Intestinal Inflammation and Tissue Repair by Environmental Factors. Thank you.
Thank you very much for the kind introduction, Carla. I shall try and share my screen now.
Yeah, very soon we can start doing that. But as you know, before we start, we really love to learn more from our speakers and learn about their experience and their thoughts. So as you share, which is great, I can see your first slide, I would like to ask you if you could share with us, what are the scientific discoveries that inspired you the most?
Well, as you said, I was fortunate to work for some time in the Basel Institute. And when I joined there in 1985, that was just the time when Antonio Lanzavecchia had discovered how B cells capture antigen, internalize it, process it, and elicit T cell help. That was an exciting period. And of course, in those years also that the discovery of the T cell receptor had a huge influence, not just on my work, but every immunologist was affected by that. And perhaps a bit later in Mill Hill, our Th17 era was really very strongly influenced by a seminar Dan Koa gave in Mill Hill, where he reported the existence of this novel subset of CD4 T cells that was involved in inflammatory diseases such as rheumatoid arthritis and brain diseases. And at the time, it wasn't possible to culture these cells in vitro. And that really spurred one of my postdocs to look into that. And he eventually discovered the differentiation factors that allowed making these cells in vitro, which of course accelerated a lot of the research then. But without Dan's seminar, we might never have heard of these cells. So this was hugely influential for us.
That's wonderful. Thanks for sharing that. So nice to hear from people that have experienced those he discovers firsthand, and how important it is communication, right? That can really help other labs, you know, contribute as well and move the field forward. That's wonderful. Thank you, Gitta. We can see your first slide perfectly fine. So let's get started.
Shall I kick off?
Yes.
Yeah.
Thank you.
All right. So you have seen my title. Let's just move on. See, and I'm already.
You might need to use in the bottom. You might need to go with the next one, with the one on the that one.
Yeah,
yes.
That works.
Perfect.
So just to this is a concept familiar to many of us, of course, that the state of our gut really has huge implications for general health. And inflammatory diseases in the gut that can culminate eventually in cancer formation are not just influenced by our genetic profile, which of course shapes host metabolism and immunity, but also by how we eat, and that in turn influences the state of our gut microbiome. So it's a complex system that has huge implications for general health. And to just orient you there in the intestine, small intestine, the follicle-associated epithelium, and the colon is a bewildering collection of different cell types that interact, and it's actually a prime example of cooperativity, because the cells of the hematopoietic system that are found in the gut crosstalk with the epithelial barrier and with the luminal and interepithelial commensal microbiota. And the interactions between all these three contribute to the maintenance of gut homeostasis. But what we got very interested in is also superimposed on these interactions are reactions to environmental triggers. And in that sense, there is a very important ligand-dependent transcription factor, which is called the Aryl hydrocarbon receptor. It's a member of a family that share certain features like the basic helix, loop helix domain and two pas domains. These are all sensors of environmental change, such as circadian rhythm, hypoxia, or in the case of AHR, the xenobiotic response. And what was really attractive to us focusing on this is that the AHR is an ideal molecular entry point into understanding the impact of environmental factors on immune and other interactions in the gut. You may wonder how we got started with this, and it was really a completely serendipitous discovery. When we had found the differentiation factors that allowed generation of Th17 cells in vitro, we put all these CD4 T cell populations on microarray at the time and looked specifically for cytokines and for transcription factors. And there we found specifically expressed in the Th17 subset, apart from raw gamma T, which we left alone because Dan Littman was working at it at the time. We found this Aryl hydrocarbon receptor. And we found out that what it does in Th17 cells is induce the cytokine IL-22 upon exposure to activation of AHR with ligand. So you see here in a wild type mouse, addition of an AHR ligand that leads to AHR activation gives you this population of IL-22 producing cells. AHR This is very sensitive, this thing. Sorry. AHR deficient cells, on the other hand, although they can differentiate to Th17 cells, perhaps a bit less efficiently, they are unable to make IL-22. So subsequently, we worked our way through various immune cell populations, but we found out that AHR seems to be particularly important at what we would call barrier sites of the body, like the skin, the mucosal immune system, and the gastrointestinal barrier, and the respiratory system in the lung. And I had mentioned to you ligands. It's a ligand-dependent transcription factor, and was studied for decades in the toxicology field in the context of xenobiotics or environmental pollutants, such as, actually I should activate my pointer, maybe that would work. The prototypic ligand is dioxin, TCDD, and various environmental pollutants. Benzpyren is your barbecue-associated environmental pollutant. So this had been studied extensively, but it never quite fitted with the fact that AHR is evolutionarily conserved from invertebrate onwards, and therefore unlikely to just be responsible for dealing with environmental toxins. And in the last 10 years, a lot of effort was made to identify so-called physiological ligands, which are largely indoles, tryptophan-derived components, dietary indoles, such as these components and dim that are derived from broccoli, like cabbages, green cruciferous vegetables, or a ligand we're using a lot in our experiment, shortened FITS, which is a tryptophan metabolite formed by light exposure. And then also it became clear that there are many of our commensal bacteria are capable of metabolizing tryptophan to AHR activating ligands. Now, to come back to AHR expression in the gut, we made use of a reporter mouse, an AHR tomato mouse, which allowed us to more easily look at protein expression of AHR in different cell types. And as you can see, there is in B cells, and the standard number of T cells are low in AHR expression, but it is Th17 cells, particularly gamma delta T cells, express high levels. And the highest levels are actually in endothelial cells and also epithelial cells and some of the myeloid lineages. So it is strongly and widely expressed in the gut. Now, expression per se doesn't mean many things, because AHR acts once it is activated by ligand, and that pathway is quite critical. So AHR normally sits in the cytoplasm, complexed with chaperones that keep it there. And upon ligand exposure, it translocates to the nucleus, and there dimerizes with the cofactor ARNT, and that complex is then capable of binding to DNA and act transcriptionally, and it binds to so-called dioxin response elements. And what was studied for extensively for a large number of years is the immediate downstream consequence of AHR activation is the expression of a number of negative regulators, such as the AHR repressor, which is thought to compete with AHR for the cofactor ANT, and then 3 enzymes of the cytochrome P450 family of drug metabolizing enzymes, whose function it is to metabolize the ligand and thereby terminate the stimulus. And this is a very important feature of AHR activation. In fact, almost as soon as it is activated, the whole battery of negative regulators is set in motion. And it is thought that physiological ligands are very rapidly metabolized and therefore only give transient activation, whereas things like the prototypic agonist dioxin is barely metabolized at all, and it's now becoming clearer that perturbation of physiological functions by abnormally prolonged AHR activation is probably at the bottom of the negative effects of many of these xenobiotic ligands. So what we have to emphasize is AHR activation is ligand dependent. There are a few exceptions which are complicated, but this means AHR expression per se doesn't tell us if it is active. And secondly, another important point is that substantial effort goes into ensuring transient activation. And we can therefore say that prolonged activation may be as much a problem as no activation. And because it is transiently activated, this makes it actually quite difficult to detect AHR activation physiologically. And we, in order to do this, we made use of a CYP1A1 Cree fate reporter that was generated by collaborators in Dundee. So Cree was put under the control of CYP1A1. So when AHR was activated and this important downstream target gene was activated, we got Cree expression and the mice were crossed to Rosa reporter mice. And what we can see on the top is the expression in endothelial cells characterized by VEGFR2. In a standard, it's activated very highly in these endothelial cells. But you see when we remove the 3 enzymes that are responsible for degrading the ligands, the activation looks even stronger. And it's easier to see in the gut. When we look at gut epithelium, there's actually relatively minor reporting in the normal CYP Cree fate reporter. And this is due to the fact that the other 2 CYP enzymes are still active, can degrade the ligands very rapidly, and therefore there isn't enough time for Cree expression to work efficiently. If we remove these metabolizing enzymes, you can now see that the gut lights up like a Christmas tree. It's very very strong reporting of AHR activity. So what happens in the gut when the AHR pathway is dysfunctional? There's a lot of work from a lot of groups in the meantime, and it turns out that AHR function is really very critical in the gut. Without AHR, the gut loses a whole set of particular immune cell types, such as intraepithelial lymphocytes and the innate lymphoid cells of the ILC3 subset, both of which are dependent on AHR triggers for their survival. AHR loss also leads to disbalance of the commensal bacteria with an increased bacterial load. There's increased susceptibility to infections. We lose IL-22 because we lose the ILC3 subset, and Th17 cells without AHR also can't make IL-22. And this leads to chronic inflammation due to barrier disruption, and that in itself is an increased risk of colon tumorigenesis. Now, I just want to highlight the two main tools that we are using, that I'm talking about in the rest of my talk, which are mice in which we put the CYP101 expression under control of the ubiquitously active ROSA26 promoter. So it's not anymore controlled by AHR, but constitutively active all the time. We have it in versions that allow either whole body overexpression or via LOXP sites, tissue-specific overexpression. And what happens is, because this metabolizing enzyme is on all the time and enzymatically active, we get constitutive degradation of AHR ligands, which results in reduced AHR signaling and actually phenocopies AHR deficiency. So we call them sometimes CYP, ROSA CYP mice, or ligand deficient mice. And the second group of mice are mice deficient a floxed AHR locus crossed to different Cre lines, and the main focus here is on willing Cre to remove AHR expression from epithelial cells. So we have AHR deficient mice and ligand deficient mice. Now, to look at the physiological consequences of AHR mutations in the gut, in fact, what is important to note is that under steady state conditions, there isn't much deficiency notable as long as you keep your mice in strictly SPF conditions. AHR mice would not do well in dirty conditions, but if the microbiota is strictly controlled in the environment, you don't see much wrong with them. However, if they are infected, and in this case we often use Citrobacter rodentium, a gram-negative bacterium, which mimics human infection with enteropathogenic E. coli. So when AHR deficient mice are infected with Citrobacter, they really are highly susceptible and can't withstand this infection, because IL-22 is a very important factor in the defense here, and that is missing in these mice. So what we get is here, and the top you see staining for Citrobacter in red. So in a wild type mouse on infection, Citrobacter sort of reaches the epithelium but doesn't get any further, and it's very rapidly controlled and eliminated. Whereas in an AHR deficient mouse, or mouse that has excessive ligand degradation, you can see that Citrobacter penetrates all the way through the epithelium to the crypt stem cells. And what you get here is very high levels of CFU, of course, in the colon, but in contrast to wild type mice, these bacteria disseminate to the liver and the spleen, and we see no production of IL-22, and these mice rapidly succumb to this infection. Now, having these, what we call ligand deficient mice, which however have an intact AHR pathway, gives us the chance to check whether we can rescue such mice by simply saturating the enzyme, either giving an enzyme inhibitor or giving more substrate. So what we do is we feed mice on a diet supplemented with this properly-derived indole-free carbinol component for 4 weeks. And it is known that in the conditions, low pH in the stomach, this undergoes an enzymatic condensation reaction leading to 2 major metabolites, nothing to do with microbiota. In this case, it's a purely chemical reaction, and very potent agonist, and this is a CYP inhibitor. So after 4 weeks of mice being on this diet, we infect them again with Citrobacter rodentium. And you can see the consequence that such mice that were exposed to these dietary AHR ligands are now protected. They can survive this infection, whereas otherwise mice will succumb by day 12 after infection. We can see also that they can now control their bacterial load again, as otherwise with the mice that are on the control diet just don't make it longer than that. And you can see in the histology that while control mice again show the spread of Citrobacter throughout the epithelium, with the I3C diet that is prevented. In this case, that rescue was very much dependent, at least in the early phase, on the presence of IL-22. And if we treated mice that were under the dietary AHR ligands with neutralizing antibodies to IL-22, they again succumbed to this infection. So IL-22 plays an important role here, but it does not, as I will show you in a moment, does not do everything. This just shows also that the I3C diet will very much reduce the histology score, and we can rescue IL-22 expression again, whereas IL-17 and gamma interferon expression are not affected in this case. And we have seen that we get the same rescue effect whether we have the pathology all over the body, whether mice overexpress CYP in every tissue in the body and every immune cell in the body, or when the overexpressing enzyme is restricted to gut epithelium. And interestingly, if gut epithelial cells alone overexpress or have overactive CYP1A1 and degrade ligands they come in contact with, this starves the whole intestinal mucosal immune system of AHR ligands and leads to loss of ILC3 and loss of IL-22. So what we were arguing is that intestinal epithelial cells and their state of AHR ligand degradation is really important, and these cells act as gatekeepers for the supply of ligands to the intestinal immune system. Right, go to the next. So as I said to you, if we look at the ligand overactive mice, it seems to be that IL-22 really does the trick when we can substitute that and boost its expression with the rescue through diet. These mice can withstand Citrobacter infection. But interestingly, that doesn't work through the whole time course. If we look at mice that lack completely AHR just in intestinal epithelial cells, such mice cannot respond to ligands, so we can't rescue them. They cannot induce CYP1A1, so they wouldn't degrade anything. But in contrast to the ligand overexpressing mice, they have a completely normal immune compartment. So they have IL-22, they have normal numbers of ILC3 and Th17 cells. However, when we look in Citrobacter infection, they're equally susceptible to this infection. So IL-22 is not enough to rescue them from the problem. And hang on, where am I? So we now know that mice with dysregulated AHR are also more susceptible to tumorigenesis. And this links with gut inflammation. Of course, it's also known that many gut infections can set you up for subsequent tumor development. To test this here, we used a DSS-AOM system, which is a standard model of inducing inflammation connected tumorigenesis. And what was very clear that mice deficient in AHR in the epithelium only, as well as mice with a hyperactive CYP in the epithelium, very rapidly develop multiple tumors. We had to actually scale down the DSS dose because AHR deficient mice are so susceptible. And under these conditions, wild type mice never developed any tumors in that time frame. So the gut inflammation situation in mice that don't have AHR in the epithelium is really setting them up for tumor formation. And what underlies that is a faulty regulation of the Wnt pathway. And here we found out that two important negative regulators of Wnt signaling, which deal with threshold and Wnt expression here, ZNRF3 and RNF43, are directly regulated by AHR, and they're downregulated in intestinal epithelial cells from AHR deficient and ligand deficient mice, as you can see here. And in chip analysis, they were directly targeted by AHR. And as a consequence of this defective negative feedback, what you get is a really hyperactive Wnt pathway. So you can see here, compared to the control, AHR deficient and ligand deficient mice have less expression of these two negative regulators, but as a consequence, much increased expression of Wnt targets such as AXIN2, c-MYC, and ephrin-B2. And we also get increased beta-catenin expression in these mice, all of which are hallmarks for the formation of cancer development. So we have now a situation where we know that AHR selectively in intestinal epithelium influences important homeostatic barrier mechanisms that prevent dysregulation and inflammation to lead to tumorigenesis. And we have now I want to show you now more recent data focused on by Kathleen Char in the lab with the help of Murali Maradana, who wanted to pick up where this whole dysregulation originates that in the end leads to increased colon cancer formation. And they discovered that AHR is really important in regulating injury repair and differentiation in intestinal epithelium. That's what I'm showing you now. So the DSS model of epithelial damage has been very well studied with very clear kinetic understanding, and is divided in several phases. There's an injury phase, ulceration phase, a repair phase, and then around day 30 or so in the wild-type mouse, you will re-establish homeostasis and have regenerated an intact epithelium. The injury itself causes de-differentiation of mature epithelial cells to a fetal-like, stem cell-like state, which is dependent on the transcriptional regulator YAP1, and can be visualized by the expression of this fetal marker SCAR1. What you also get is loss of differentiation markers for mature epithelium. It leads to cell cycle re-entry and a reacquisition of stemness. These are all normal processes that happen when tissue is injured and needs to be regenerated. Now, when Kathleen looked at the different phases after DSS injury in either on top wild-type mice and on the bottom mice lacking HR in the epithelium, you can see that the initial phases are fairly similar. Of course, one could say the injury phase is exacerbated in the HR deficient mice, but you do get very clear expression of this fetal remodeling expression SCAR1 also in wild type epithelium. But over time, up to 30 days, the wild type epithelium now looks completely normal, whereas the epithelium from HR deficient scenarios is still completely dysregulated, and the injury process is continuing. So actually these mice cannot stop the regenerative process anymore, and they keep on remodeling. You can see this quantify this by the expression of SCAR1. What also happens, you get mucosal thickening and disorganization of crypts, which just doesn't terminate anymore in HR deficient epithelium. And what you can see in addition is, again, on top wild type, on the bottom HR deficient epithelium. During that injury, you lose mature epithelial cells such as goblet cells. That's visible both in wild type and HR deficient mice. But eventually, the wild type mice regain their normal numbers of mucus producing goblet cells, whereas this doesn't happen for HR deficient epithelium. And you can also quantify that by expression of mature epithelial markers such as MAC2 and KIT20, which are underexpressed in oh no. Oh, yeah.
That's good.
I'm back, no? Okay.
Yes, you're back. It's not there. That's perfect.
Yeah. So to sum this up, HR deficiency in gut epithelium leads to epithelial cell intrinsic defects such as failure to resolve injury repair. I didn't show you, but there's also metabolic rewiring defects. They don't switch on oxidative phosphorylation that normally happens during differentiation. And simply the differentiation step does not occur. So in order to get at mechanistic details of what's going on there, we went to colon organoid cultures. in some way, it is now clear that organoids, the whole process of ripping stem cells from their normal environment and setting it up in these organoid cultures, represents a kind of injury. And this tissue dissociation recapitulates aspects of tissue injury, which results in upregulation of this YAP1-driven reprogram process. So what we do is we culture these colon organoids either in Wnt factors and as a stem cell state, or we withdraw Wnt and allow them to differentiate, where we can then look at termination of this repair process and maturation and differentiation. And in order to get at, as I said, the molecular mechanisms underlying this, we resorted to multi-omics RNA-seq analysis of stem cell and differentiated organoids, ATAC-seq to look at the epigenetic situation in stem cell and differentiated organoids, again between wild type and HR knockout, and ChIP-seq to get at the direct targets of AHR. Now, what we first saw in looking at RNA-seq conditions is what happens is that HR deficient colon organoids have an increased fetal-like signature expressing at very high levels so-called YAP targets, and YAP is I referred to it before, is a transcriptional regulator that activates genes, gene transcription for proliferation and interferes with, suppresses apoptotic genes. So this is hugely upregulated in an HR deficient scenario, and this is a number of YAP targets that are validated there at being overexpressed. So the whole epithelium is poised towards excessive regeneration and proliferation. And if we take organoids in the stem cell state and expose them to AHR ligand by activating AHR, you can actually see that this regenerative abnormality of course, only in the ligand deficient scenario, is downregulated, and the target genes are downregulated. So really, HR activation helps to terminate that repair state. And that follows from what we first seen, that in the absence of AHR, there is just chronic and continuous repair. So now, how does AHR influence YAP activity? We didn't have evidence of any direct effect on YAP differential expressions and so on, but knowing that rewiring of transcription networks during differentiation requires remodeling of the epigenome, we resorted to ATAC sequence to look at how the state of the chromatin in these two situations between wild type and HR deficient organoids looks. So if we look at the heat map of differentially accessible peaks in the stem cell state, you see sort of in blue are the accessible or open chromatin configuration. There is a difference between wild type and HR deficient, but it's not quite so evident as it is here in the day 4 Wnt withdrawal differentiating conditions, where really many more loci remain in an open configuration in the absence of AHR. And there's access and gain in 81% of these loci and in only 18%. So what actually happens is that there is increased transcription factor footprinting. If you look at which transcription factors have access to these remaining open chromatin configurations, it is tiered and ETS motifs, and YAP exerts its transcriptional control via tiered mediated activation of enhancers, and the ETS family members collaborate in this process. So increased binding of transcription factors that dictate the balance between regenerative and pro-differentiation programs tips this into the pro-regenerative but anti-differentiation direction in the HR knockout organoids. Now that was that is one side of coin, explaining how this regenerative response fails to be terminated. The other effect, as I told you, is failure to differentiate. And if we again look in the RNA-seq data, we see that there is a strong reduction in markers characteristic for mature epithelial cells, tuft cells, goblet cells, and enterocytes. And this is also shown in the validation. And we can actually also see that in human colon organoids, where when we block, in this case, HR activation with an antagonist, we interfere with the expression of mature epithelial differentiation. And then we looked at an overlap between ChIP-seq and RNA-seq and found a certain proportion of genes with increased expression, such as CDX2, HNF1 alpha, very important in differentiation, and a similar proportion of genes with this decreased expression in the ChIP-seq, SOX9 and proto-oncogenic targets. And what we could see is that we can actually see that CDX2 is a direct HR target. You see here the ChIP peak, and this sits in an open ATAC-seq open chromatin region. And we can also see it by staining, that CDX2 staining in the HR deficient situation is decreased, which explains the problems with differentiation. Then SOX9 is important for self-renewal and repair, and represses CDX2 and MAC2, and also YAP is linked with SOX9. So we know that YAP1-driven SOX9 expression is a critical event in acquisition of cancer stem cell properties, and SOX9 is normally repressed by HR. Again, you see the peak here in the open chromatin regulation, but in contrast to wild type, you see increased SOX9 expression, so increased propensity for oncogenic transformation in HR deficient organoids. So just to wrap this up, HR is really important in regulating regenerative and wound healing responses in adult intestinal epithelium, and is required to terminate the regenerative program. That means it needs to repress SOX9, shut down access to YAP targets. It promotes and regulates stem cell differentiation by direct positive effects on the CDX2 enhancer. And of course, dysregulation of these processes are major contributors to inflammatory disease and cancer. So now in the last few slides, I just want to come to a persistent, perplexing issue we have in the field. So we have now shown that HR activation under physiological conditions is beneficial and really important in gut homeostasis. However, HR is linked with all these negative effects, for dioxin, for instance, profound consequences on vertebrates for developmental endpoints. And many of these effects actually happen on processes like differentiation. So what is the difference? One thing that also has to be mentioned, because it's a big problem in the toxicology field, it's been shown largely through in vitro data that metabolism by CYP1A1 can generate toxic metabolites that function as DNA adducts and directly initiate cancer. So this all doesn't sound very beneficial, but it looks as if this CYP-mediated so-called metabolic activation may not be common under in vivo conditions. Experiments have been done with CYP knockout mice, which suffer more from not being able to metabolize than from getting tumors. But in general, exposure to AHR ligands that are poorly metabolized, environmental pollutant exposure is a common occurrence in the modern world. And how does this impact on processes such as tissue repair? And Kathleen did one preliminary experiment that is still work in progress, doing the same kind of DSS experiment in the context of particulate matter, which was collected from environmental car, diesel exhaust, and so on the Marylebone Road in London, where you wouldn't want to walk along too much. And what she can see, these are wild-type mice in both cases, but exposure to these pollutants under DSS really causes almost, it mimics AHR deficiency. So again, there is failure to repair properly in this setting. So how can we reconcile the detrimental effects of AHR activation by these xenobiotics with the beneficial effects by endogenous ligands? And that's what we're looking into now. Are the downstream pathways different? Probably not. The duration of signaling is clearly different. And perhaps one hypothesis could be that prolonged activation leads to degradation of AHR so that it's not there in the critical time window when it would be needed. But this is all I wanted to say, and I want to finish with acknowledging firstly my lab in not so good times and very recently on an excursion to Greenwich, which was much better. My lab and current and also past lab members, collaborators, and the quick technology platforms that help us with all the various technology support, and our funders. And thank you for listening.
Thank you, Gitta. That was wonderful. Thanks for sharing these very important discoveries, and especially your very methodic approach to try to understand the role of AHR from the organismal level to the molecular level. Thank you so much. Before we finish, I would like to again share my slides and remind sorry, I'll have to go through this 1 second, and remind everybody how we can ask questions. So as a reminder, we ask questions through Twitter. So if you go to Twitter, you will already find a tweet that says, ask questions to Dr. Gitta Stockinger here. And please remember to reply to that tweet with your question and mention Global Immuno and the ImmunoSpeaker account, which Gitta is going to use to answer these questions. So Gitta, thanks again. That was fantastic. We really appreciate it.
Carla, thank you very much for your kind introduction and help in between when I lost my...
Everything was super clear. Everything was wonderful. Thank you, and goodbye, everybody. We look forward to seeing you next week. Join us for Feng Xiao's talk. Bye, everybody.
Bye.